Hello. Today I will be discussing the measurement of eicosanoids and their urinary metabolites using mass spectrometry. First, what is an eicosanoid? Eicosanoid is an umbrella term used to describe any oxygenated derivative of a 20-carbon essential fatty acid. A few examples of 20-carbon essential polyunsaturated fatty acids are shown here on this slide. For the purpose of today's talk, we'll be focusing on arachidonic acid, depicted here in the middle. This has 20 carbons and 4 carbon-carbon double bonds. So eicosanoids have been studied for over the past 80 years. It was first in 1930 that two American gynecologists observed that uterine tissue relaxes or contracts upon exposure to seminal fluid. A few years later, von Euler identified the responsible compound as a lipid-soluble acid. Because he thought this compound was coming from the prostate gland, he termed this molecule prostaglandin, but the structure was not yet known. In fact, it was nearly 30 years later before the structures of prostaglandin were elucidated. And then, at that point, these were determined to be not just one compound, but a family of compounds. In 1971, Vane, Smith, and Willis determined that the drug aspirin blocks prostaglandin biosynthesis. So this enabled the discovery of the enzymes that actually form these molecules. Prostaglandin synthase, also termed cyclooxygenase or simplified COX, were identified as the enzyme that forms prostaglandins. The intermediate, PGG2, was determined, and other products of the cyclooxygenase enzyme, such as thromboxane B2 and prostacyclin, were all discovered in the mid-1970s. Nearly 20 years later, it was discovered that it was not just one enzyme, but two cyclooxygenases that are responsible for the formation of these compounds in vivo. Shown on this slide is the pathway that we now understand for prostaglandin formation. Arachidonic acid is stored in the body in cells, esterified in tissue lipids, such as phospholipids. Physical or chemical stimuli can activate the cells to cause an influx of calcium, which will release arachidonic acid from the phospholipid membranes by the action of enzymes called lipases or phospholipases. Arachidonic acid is then a substrate for the enzymes cyclooxygenase. As we discussed previously, there are two cyclooxygenase enzymes, COX-1 and COX-2. The initial intermediate that is formed is a bicyclic endoperoxide compound with a hydroperoxide group at the 15 position. This intermediate is short-lived and is termed prostaglandin G2. The cyclooxygenase goes on to reduce the hydroperoxide at the 15 position to an alcohol to yield prostaglandin H2 or PGH2. This is the gatekeeper prostaglandin. The bicyclic endoperoxide functionality of PGH2 is then transformed to yield a number of different prostaglandins. These are all generated from prostaglandin synthase enzymes. Since their discovery, prostaglandins have been shown to exert a number of different biological activities, and they exert these biological activities through G-coupled membrane protein receptors. As you can see, the prostaglandins are formed in many different cells in the body in different regions, and they act locally in these regions, exerting a number of different activities. Prostaglandin E2 is the most abundant prostaglandin generated in vivo. It is known as a bronchoconstrictor, a vasodilator. It induces fever as well as pain. In the bone, it can induce bone resorption and is also involved in the labor process through cervical ripening. Importantly, PGE2 is also known to exert potent inflammatory effects when it's generated from cyclooxygenase 2. In the mid-1970s, at the same time the biology and the structure of the prostaglandins was being studied, Hamburg and Samuelson described an enzyme in platelets that was separate from the cyclooxygenase that inserted molecular oxygen at the 12 carbon of arachidonic acid. This type of enzyme was termed a lipoxygenase. In 1975, steroids were proposed to block formation of prostaglandins, but the method of action was different than that to NSAIDs. These hints led to the discovery that there were multiple enzymes that could oxidize arachidonic acid to other compounds. In 1976, Samuelson and colleagues discovered that arachidonic acid can be oxygenated at carbon 5 in rabbit leukocytes. And they went on to discover that this oxygenated product at carbon 5 can be transformed to a compound they termed leukotriene B4. In 1979, they also elucidated another set of compounds forming from lipoxygenase intern. In the mid-1970s, along at the same time that the structure and identification of the prostaglandins was being elucidated, Homburg and Samuelson described an enzyme in platelets that would insert molecular oxygen at the 12 carbon of arachidonic acid. A couple of years later, they found that arachidonic acid could also be enzymatically oxygenated at carbon 5 in rabbit leukocytes. These observations led to a series of studies that identified a new class of compounds that could be generated from the oxidation of arachidonic acid, termed leukotrienes. Importantly, in the early 1980s, the Nobel Prize in Medicine was awarded to several investigators for their discoveries related to eicosanoids, including prostaglandins and leukotrienes. This slide shows leukotriene formation. To generate leukotrienes, arachidonic acid is acted upon by an enzyme complex of 5-lipoxygenase and its activating protein flap. The initial product of arachidonic acid oxidation by the 5-lipoxygenase is this compound termed 5-HPEAT. It's a hydroperoxide, which is 5-hydroperoxide eicosatetraenoic acid. This is converted by the enzyme LTA synthase to yield the short-lived intermediate leukotriene A4. This is our gatekeeper leukotriene, which can either be transformed by LTA hydrolase to yield leukotriene B4 or act upon by leukotriene C4 synthase to yield a family of compounds termed cysteine leukotrienes. LTB4 and the cysteine leukotrienes act through G-coupled membrane protein receptors, just like the prostaglandins. 2 receptors for each leukotriene have been identified. The leukotrienes are known to be heavily formed in the lungs and are important in the allergic response. They recruit immune cells as well as are bronchoconstrictors. In the early 1980s, a third class of compounds was shown to be generated from the oxidation of arachidonic acid via enzymes. This discovery was made by Capdevila and colleagues, and it was shown to be formed from the oxidation of arachidonic acid by cytochrome P450 enzymes. Cytochrome P450 enzymes are important in the metabolism of many compounds in the body, including pharmaceuticals. The products that are generated from the oxidation of arachidonic acid by cytochrome P450 are monooxygenase products, meaning that they have one oxygen inserted. There are different classes of compounds that can be generated, including the epoxy eicosatetraenoic acids, shortened to be called EETs. These are formed by 2C and 2J enzymes. Alternatively, CYP4A enzymes oxidize arachidonic acid to the 20-heat compound. The family of EETs can be metabolized by soluble epoxide hydrolase to yield dihydroperoxy compounds termed diheats. Receptors for these molecules have not yet been identified, such as prostaglandins and leukotrienes, but these molecules do exert potent biological activities. 20-heat, for example, is involved in hypertension and the pain response. The EETs have been shown to decrease hypertension as well as insulin resistance. In recent years, soluble epoxide hydrolase inhibitors have been made due to the beneficial effects of EETs being able to decrease blood pressure and insulin resistance. However, it has also been shown that EETs are angiogenic and cause cell proliferation and may play a role in cancer. So caution must be used when using soluble epoxide inhibitors to treat patients. What we've discussed so far is all enzymatic oxidation of arachidonic acid. Importantly, arachidonic acid, due to its structure and the 4 carbon-carbon double bonds, can also be oxidized by non-enzymatic lipid peroxidation. The general pathway of lipid oxidation is shown here. So a free radical. Free radicals can include compounds that are formed in the body, such as superoxide, and they can also be generated endogenously from environmental stimulus, even something as simple as going out in the sun. A free radical can abstract a hydrogen atom at a bisallylic position in between carbon-carbon double bonds. Once a hydrogen atom is abstracted from the lipid backbone, the lipid radical can react with molecular oxygen to yield lipid hydroperoxides. Lipid oxidation has been extensively studied, and there are several different types of compounds that can be generated. Some of the initial products that are formed are hydroperoxide compounds. If you recall, these are some products that can also be generated from enzymatic oxidation of arachidonic acid. Hydroperoxide compounds can be reduced to generate heats. The molecules can undergo further extensive oxidation to yield secondary oxidation products such as isofurans or isoprostanes. And in addition, lipid hydroperoxides can undergo fragmentation to yield lipid aldehydes. As you can note, trying to actually study these molecules can get complicated pretty quickly due to the fact that these compounds are similar in structure to enzymatic oxidation products of arachidonic acid. Now, mass spectrometry has played a crucial role in the identification and characterization of eicosanoids and their biosynthetic pathways for over 50 years. If you do a literature search on eicosanoids and mass spectrometry, you can find review articles on using mass spectrometry in this way as early as 1975. So mass spec has been used to look at these compounds since the 1960s and has played a crucial role in their structural elucidation, as well as determining at what levels these compounds are generated in vivo. But if we look at just the snapshot of enzymatic and non-enzymatic oxidation products on this slide, you can see that many compounds can be generated from different pathways, and their structures are very similar to each other. So very quickly, the analysis gets quite complicated. In the early days, gas chromatography mass spectrometry was used to measure eicosanoids as biomarkers in vivo. There are still many labs today which use GCMS to study these molecules due to its sensitivity and ability to measure very low levels of these compounds. In more recent years, liquid chromatography mass spectrometry has been used due to its ease of use compared to the derivatizations that are needed for GCMS. Immunoassay methods are also available to quantify eicosanoids. However, due to the structural similarities between these molecules, there's much cross-reactivity associated with the immunoassay quantification of these molecules. In general, immunoassay methods are always quantify levels significantly higher than mass spectrometry. In addition, immunoassay quantification and mass spectrometry quantification often do not correlate with each other. Thus, mass spectrometry is the method of choice to quantify these compounds. But when we think about eicosanoids as biomarkers and how to measure these using mass spectrometry, we need to consider the lessons of the past and take in special considerations. First, eicosanoids are generated at very low levels in vivo, so sensitive quantification and specific quantification is a must. Importantly, it's important to note that prostaglandins are rapidly metabolized. For example, after injection of PGE2 into an animal, 3% of it remains in the blood after 1.5 minutes. So trying to quantify prostaglandins as well as other eicosanoids in the blood is not feasible or recommended because the levels that are generated are very transient and not reflective of systemic production of these molecules. Some species are unstable when a sample is taken and left at room temperature ex vivo, so sample handling is very important. And further, there are many isobaric species. So for example, two of the main parent prostaglandins, prostaglandin E2 and prostaglandin D2, both have a molecular weight of 352. A search of eicosanoids on the Lipidomics website, shown here at www.lipidmaps.org, show that there are more than 24 different types of molecules that have this same molecular weight of 352. And please note that this is 24 different types of eicosanoids. This does not include different isomers of these same compounds. So what I hope to share with you today in the next half of my talk is a couple of stories that we've learned along the way when studying eicosanoids and looking at the utility of them as biomarkers. When preparing this talk, I came across this quote that said that the universe is made up of stories, not of atoms. As chemists, we believe, of course, that the universe is made up of atoms. But when thinking about how to quantify compounds, it's useful to tell a couple of stories to indicate the types of issues that we need to consider when developing a mass spectrum method to measure these molecules. The first story that I would like to tell is of a prostaglandin D2 metabolite that wasn't. So we're going to go back and look at the formation of prostaglandins. This shows the family of different compounds that are generated from the oxidation of arachidonic acid by the cyclooxygenase. Of importance to this story is prostaglandin D2. So in the mid-1980s, researchers by the names of Jack Roberts and Jason Morrow were interested in PGD2 because this was known to be activated in mastocytosis, a disease of the mast cells in vivo. And what these researchers were trying to do is identify urinary metabolites that could be quantified. In order to start this story, we need to go back to this slide showing the formation of prostaglandins. Now, arachidonic acid, as we mentioned, is oxidized by the cyclooxygenase to a family of prostaglandin compounds. Of importance to this story is prostaglandin D2. In the mid-1980s, researchers by the names of Jack Roberts and Jason Morrow were interested in prostaglandin D2 because it was known to be increased in mast cell activation. And in particular, they were interested in a disease called mastocytosis. Now, prostaglandins, as we mentioned on the previous slide, are rapidly metabolized. So it's recommended that in order to quantify prostaglandins, you look at their metabolites and not at the molecules themselves. So these researchers were looking for a metabolite of prostaglandin D2 that you can measure in the plasma to indicate formation of this molecule in vivo. And what they determined was that prostaglandin D2 can be metabolized by 11-keto reductase to yield 9-alpha 11-beta prostaglandin F2 alpha. Now, importantly to note, 9-alpha 11-beta prostaglandin F2 alpha is an isomer of PGF2 alpha, which is also a product of the cyclooxygenase pathway. What they observed when they were studying this metabolite by gas chromatography mass spectrometry was that there were at least 16 different prostaglandin F2 stereoisomers that were generated from the metabolism of PGD2. And so in a publication in 1988, they made the conclusion that PGD2 is a very labile compound in vivo, and that it undergoes extensive isomerization, after which reduction by 11-keto reductase yields a family of stable isomeric PGF2 compounds as metabolites. This slide here shows a chromatogram of the different peaks that they observed in the GCMS that they attributed to prostaglandin D2 metabolism. They had a sample of fresh plasma, and they quantified PGD2 metabolites in this sample at levels of 40 picograms per mil, using a deuterated internal standard of 9-alpha 11-beta PGF2 alpha. They put that sample in the freezer and stored it at minus 20 degrees Celsius for 6 months. When they went back to reanalyze this sample, they realized that levels had increased dramatically, up to 2,000 picograms per mil compared to 40. So they took a step back and said, what in the world is happening here? And what they determined that actually the peaks were not representative of metabolites of PGD2, but were non-enzymatic oxidation products of arachidonic acid that were being generated isomeric to prostaglandin F2 alpha. Only the small peak in the chromatogram that elutes at the same retention time as the deuterated internal standard was the prostaglandin D2 metabolite, 9-alpha 11-beta. The other peaks in the chromatogram are formed from non-enzymatic oxidation. This slide shows the pathway of F2 isoprostane formation. A radical species abstracts a hydrogen atom from one of 3 positions on the arachidonic acid backbone. This lipid radical then reacts with molecular oxygen, which undergoes rearrangement and cyclization. A second molecule of molecular oxygen is added, and it yields a family of different F2 isoprostane compounds. All of these molecules have the same molecular weight and are isomeric to prostaglandin F2 alpha, the product of the cyclooxygenase pathway. A total of 64 different stereoisomers are formed. There are key differences between prostaglandin F2 alpha and F2 isoprostanes. Prostaglandin F2 alpha is formed from free arachidonic acid that has been released from phospholipid membranes. F2 isoprostanes can be generated in situ on phospholipids, so arachidonic acid can be oxidized free or when it's esterified. PGF2 alpha has side chains that are trans to one another, whereas F2 isoprostanes have cis formation side chains. Furthermore, it's important to point out for PGF2 alpha there is only one isomer formed, whereas with the isoprostanes there are 64 different isomers. Since their discovery in 1990, F2 isoprostanes have been determined to be an excellent biomarker of oxidative stress and lipid peroxidation in humans. This slide just shows a snapshot of some of the diseases in which F2 isoprostanes have been shown to be elevated. So what have we learned about using F2 isoprostanes as a biomarker? We know that there are normal levels, which are in all humans, and that these have been defined. And there have been multiple methods used to quantify these molecules. One of the most popular methods to quantify them is GCMS with negative ion chemical ionization. GCMS is still popular today because of the sensitivity that it allows. With the sensitivity, you're able to quantify F2 isoprostanes in plasma. It's important to note that these compounds are present in plasma only at levels of 35 picograms per milliliter. However, the drawback to GCMS analysis is that extensive derivatization is required, and the sample load for one person is only about 20 samples per day, so this is not a high-throughput analysis. This slide shows the steps that are required and the derivatizations that are necessary to be able to quantify these molecules accurately using GCMS. In more recent years, immunoassay methods have been developed to quantify these molecules. But there have been a couple studies in the literature showing that levels measured by mass spectrometry do not correlate with levels measured by immunoassay. The levels measured by immunoassay are routinely higher than that noted by GC or LCMS, in fact. And this is probably because of the many different isomers of these molecules that are generated and the fact that they are structurally similar to prostaglandins. Now, importantly, in the past 2 to 3 years, there have been more than a dozen LCMS methods for F2 isoprostane analysis generated in the literature. And these are just method papers, not papers using mass spectrometry to quantify these as biomarkers, just simply method papers. So a big problem with the analysis of F2 isoprostanes is that there is not a single method to quantify these molecules. If you measure them in one lab and go to another, you're going to get different levels of quantification. In addition, there are multiple standards available, and different labs believe in using different internal standards to quantify these molecules. One important advantage of LCMS is that it allows for isomer class separation. So using MRM transitions, you can elucidate 5 series F2 isoprostanes, 15 series isoprostanes, as well as 8 and 12 series isoprostanes from one another. This has great potential because it would lead to a different understanding of quantification of these molecules. Importantly, in most of the literature reports, most labs have focused on the quantification of just one specific isoprostane isomer. This is the 15F2T isoprostane, which is commonly referred to as 8-iso-PGF2 alpha. This was the first of the isoprostanes synthesized and the most studied molecule. However, if we just focus on the quantification of this one particular isomer, we could be missing the entire story in vivo. So what I've hoped to emphasize in this story about the formation of F2 isoprostanes is that we have to be careful when we're quantifying these molecules because there are many different endogenous compounds which are structurally similar, and we have to consider the whole picture of this class of molecules, not just one isomer over another. To wrap up my slides today, I would like to talk about another eicosanoid biomarker, which has a potential for use in the clinic. And this is a prostaglandin E2 metabolite. The story I'd like to tell you is a story from hibernation to awakening. So we'll go back and look at the formation of prostaglandins one more time, just to emphasize PGE2 is the compound that we're interested in discussing in the next few minutes. As I mentioned at the beginning of the talk, PGE2 is the most abundant of the eicosanoids generated in vivo. In particular, this is a product generated from cyclooxygenase 2 and is involved in inflammation, and has particularly been shown to play a role in cancer. COX2 is overexpressed in many different types of cancers: colorectal, lung, breast, prostate, stomach, and esophageal, among others. Importantly, prostaglandin E2 synthase is overexpressed in many cancers as well. PGE2 is also biologically active and can signal many facets of tumorigenesis, including proliferation, vascular angiogenesis, immune suppression, and reduced apoptosis. Importantly, PGE2 has been shown to mediate metastasis of certain types of cancers. It is also important to note that PGE2 metabolism is disrupted in many cancers. The first step in the metabolism of PGE2 is reaction with an enzyme 15-prostaglandin dehydrogenase. This is downregulated in certain types of cancers, thus increasing the level of PGE2 in these cells. Now, as we've already mentioned, the best way to quantify prostaglandins in vivo is to look at their metabolites. Shown here is the metabolism pathway of PGE2. As I mentioned, the first step is reaction with 15-prostaglandin dehydrogenase. This converts the hydroxy group at the 15 carbon to a keto group. The double bond is reduced, and the molecule undergoes beta oxidation as well as omega oxidation to yield the metabolite shown here. Now, this metabolite was identified in the early 1970s, and a method to quantify this molecule using gas chromatography mass spectrometry was reported in the mid-1970s. However, to quantify this molecule by GCMS, it took approximately 1 week to analyze 10 samples. So the utility and the feasibility, honestly, of quantifying this metabolite was very difficult. So for years, there was not many studies done quantifying this metabolite of PGE2. One of the reasons this might be is because the metabolite is known to degrade ex vivo when urine remains at room temperature for 90 minutes. Thus, trying to synthesize an internal standard for quantification was quite difficult and was not reported until 2004. With the internal standard in hand, it was possible to develop an LCMS method to quantify PGEM. So urine can be collected and placed immediately at 4 degrees Celsius and is stable in the refrigerator for up to 1 week. -80 is definitely recommended for long-term storage. Now, to quantify this molecule, we have to stabilize it, so derivatization is necessary. The derivatization occurs at the ketone group on the prostane ring. Methoxamine hydrochloride is used for this derivatization and can stabilize this molecule. Once the derivatization occurs, the molecule is stable to analyze and be kept at room temperature for a couple of days. The samples, once derivatized, can be placed in the -20 freezer and analyzed a few days later. It's important to note that the derivatization yields 4 different O-methoxime PGEM isomers. These isomers are generated based upon the position of the methoxy group on the nitrogen in the derivatization portion. The internal standard that is used to quantify these molecules was able to be synthesized because it was pre-derivatized, and the deuteriums are on the methyl group of the methoxy. This LCMS chromatogram shown here shows that there are 2 different peaks that are generated in the chromatogram based on the 4 different isomers of PGEM that are generated during the derivatization. The advent of UPLC technology has allowed for a much faster quantification of PGEM, going from a runtime of 15 minutes to a runtime of about 3.5 minutes. And these 2 isomers can be baseline separated very well using UPLC techniques. Importantly, when UPLC was used to separate the isomers of PGEM, there were 4 peaks that were determined to form endogenously. Now, the standard of PGEM only has 2 peaks. This has all 4 of the stereoisomers that are generated in it. So what were these 2 extra peaks? And it was determined that these were actually metabolites of prostaglandin D2 and a route of metabolism that had not previously been identified for that prostaglandin. So this slide and this chromatogram using UPLC truly highlights the need to use good chromatography when studying compounds that are complicated, such as eicosanoids, when many different isobaric species might exist. In the early days, the PGD metabolites were being buried in this LCMS analysis, but can now be separated and quantified alongside with the PGE2 metabolite. So what have we learned over the course of the past 15 years looking at PGEM as a biomarker? There are more than 30 publications in the literature. Normal levels of PGEM have been defined, and interestingly and importantly to note, males and females have different levels of these compounds. This is unique for prostaglandin metabolites. This is unique to PGE2. As would be expected, PGEM levels are reduced by ibuprofen and celecoxib, and smokers have increased PGEM. Now, the true potential in the clinic for PGEM is as a disease biomarker. So in cystic fibrosis patients, it has been found that PGEM is associated with genotype severity. In women with HIV, PGEM levels are elevated compared to women without the disease. And importantly, PGEM correlates with plasma HIV RNA levels. One of the situations in which PGEM has the most promise as a clinical biomarker is in cancer. PGEM has been shown to be elevated in numerous different cancers shown here, and is also thought to be elevated with risk of development of these cancers. In oral squamous cell carcinoma, levels of PGEM have indicated poor prognosis. And importantly, aspirin and nonsteroidal anti-inflammatory usage decreases levels of PGEM in cancer populations and is generally associated with a lower risk of development of these diseases. For the last part of the talk, I want to focus on the potential of PGEM as a biomarker in colorectal cancer. Colorectal cancer is the second leading cause of cancer-related deaths in the USA. Prevention has become a priority. Can typical aspirin doses of 81 milligrams or 325 milligrams lower PGEM enough to reduce risk? This is a question that multiple colleagues of ours are asking. Now, aspirin is the most promising chemopreventative agent because in basic epidemiological and clinical studies, it has shown promise. It is now recommended by the U.S. Prevention Task Force for people ages 50 to 69 who have an elevated risk profile of development of colorectal cancer in their family. But aspirin is associated with a risk of GI bleeding because prostaglandins are involved in the maintenance of stomach lining. So there are many questions that need to be asked before aspirin is fully considered as a preventative treatment for colorectal cancer. What is the optimal dose? And another question is that are there subgroups of people at highest risk for development of the disease? And can aspirin be of use to this group? Now, we've done multiple studies with prostaglandin E2 metabolites in colorectal cancer. Pre-diagnostic PGEM is associated with a risk of colorectal cancer as well as adenoma formation. In the Nurses' Health Study, women in the highest quartile of PGEM levels. That means that if you separate women into groups based upon their levels of PGEM in their urine, the women with the highest levels of PGEM had the highest risk of development of colorectal cancer compared to women in the lowest PGEM level group. Aspirin and NSAID use is associated with a significant reduction of adenoma risk in the women who have the high PGEM levels. But this same reduction in risk was not found in women with low PGEM levels. So the thought here is that if you have high PGEM levels, this may qualify you and identify a subgroup of people who would benefit most from taking aspirin to prevent colorectal cancer. Aspirin has been shown to reduce PGEM levels. So in 10 individuals, we did a small study, and they received 2 doses of 650 milligrams of aspirin. and this reduced PGEM by 44%. But can typical doses of 81 milligrams per day or 325 milligrams per day inhibit PGEM to a level that is associated with a clinical lower adenoma risk? So the ASPIRE trial is actually going to look at this question. So this is a trial that is centered with Dr. Andy Chan at Harvard University Medical Center. They are currently enrolling 180 subjects who have had previous adenoma. They will randomize these subjects to aspirin at different doses or to placebo. Urine will be collected at baseline in 12 weeks. A number of different history and lifestyle factors will be observed, as well as comparing prostaglandin EM levels, PGEM levels, between baseline and 12 weeks. Currently, the ASPIRE trial is enrolling 180 subjects who have had previous adenomas. These subjects will be randomized to either aspirin at 81 milligrams per day, aspirin at 325 milligrams per day, or placebo. And urine for PGEM will be collected at baseline in 12 weeks to assess if aspirin can significantly reduce PGEM to levels that might predict adenoma risk. So what I've hoped to show you today is that mass spectrometry is a powerful tool to assess endogenous eicosanoid formation. Methods to quantify eicosanoids in humans must be sensitive and specific, and it's very important to remember the eicosanoid chemistry related to the formation of these molecules. F2-isoprostanes are a promising biomarker of endogenous lipid peroxidation and oxidative stress, and quantifying the major urinary metabolites of prostaglandins is the best method to assess their endogenous formation. In particular, quantification of the major urinary metabolite of PGE2 has a significance and shows potential as a clinical biomarker for cancer in particular subpopulations, and may lead us to an area where we can study better personalized medicine for the treatment of these individuals with high levels of PGE2. What I want to show you as I leave you today with this slide is that arachidonic acid oxidation is very complex. We've only been able to look at the tip of the iceberg today. There are many classes of compounds that we're not even able to discuss due to limited time. This area has a great potential for elucidation of clinical biomarkers, but we must be vigilant in analyzing these compounds using mass spectrometry and sensitive, specific, robust methods.